This video is audio content only. It does not contain any video footage or images. If you are expecting visual content, please be aware. This is intentionally provided in an audio-only format. We recommend listening to the audio rather than watching the screen. Thank you for your understanding. Welcome to the Table of Another, the evolution of parasitic life. Far from being biological outliers, parasites represent one of nature's most successful and recurring survival strategies. In this journey, we'll explore the Red Queen arms race of coevolution, the eerie world of host manipulation, and the surprising role genomic parasites played in building the human placenta. From ancient origins to the rapid changes of the Anthropocene, we'll discover how these master survivors shape our world from the inside out. Let's pull back the veil on the hidden architects of life. Chapter 1. The Table of Another. The Evolution of Parasitic Life. If you were to select a random animal from the millions of species currently inhabiting our planet, there is a nearly 40% chance that the creature you choose survives by hijacking the resources of another. We often think of parasites as biological outliers or rare grisly accidents of nature, but the reality is that parasitism is one of the most successful and frequently repeated evolutionary gambits in the history of life. The word parasite itself carries a social history, derived from the ancient Greek parasitos, meaning one who eats at the table of another. In antiquity, this was not a biological term, but a social one, describing a person who lived at the expense of others through flattery. Today, we recognize it as a masterful survival strategy that has emerged independently across the animal kingdom at least 223 times. This number is a staggering increase over earlier scientific estimates, which suggested only about 60 independent origins. The discrepancy exists because, for a long time, our gaze was fixed primarily on the parasites that directly affect humans. When researchers began to look systematically at the broader world of arthropods, mollusks, and even jellyfish, they discovered that the transition from a free-living existence to a parasitic one is a recurring masterpiece of natural selection. Despite its prevalence, the transition to a parasitic lifestyle is a difficult jump to make because it requires a dual mastery of both a symbiotic relationship and a trophic one. A parasite must not only feed on its host, but also find a way to live in or on it for an extended period without immediately killing the source of its next meal. This delicate balance is why we see most parasitic species clustered within a few ancient and highly successful lineages. In fact, 10 major groups of parasites contain roughly 90% of all known parasitic animal species. These include the heavy hitters of the parasitic world, the parasitoid wasps, the flatworms, and the roundworms. These groups are ancient, with many having made the transition to parasitism before the Mesozoic era, more than 250 million years ago. Because these lineages are so old, it is often difficult to find the free-living relatives they originally diverged from. Biologists distinguish between two fundamental functional divisions, microparasites and macroparasites. Microparasites are microorganisms, such as bacteria, viruses, or protozoa, that reproduce directly within the host, often at a rapid pace. Macroparasites, by contrast, are multicellular organisms like worms, ticks, or lice. These typically do not multiply within a single host. Instead, they produce infectious stages, such as eggs or larvae, that must leave the host's body to find a new home. This distinction dictates how a parasite interacts with the host's immune system and how it spreads through a population. In groups like mites and flies, we can see evolution caught in the act. These groups often contain facultative parasites, organisms that usually live independently but can become parasitic if the environmental conditions are right. This flexibility is often found in species that already exploit ephemeral or unpredictable resources, such as scavengers that feed on decaying matter. For example, some species of flies have evolved to parasitize frogs and toads. The female fly lays her eggs on the skin or inside the nostrils of the amphibian. When the maggots hatch, they eat their way into the host's tissues. Once the host dies, the larvae finish their development and emerge as free-living adults. This protelean lifestyle, where the juvenile is a parasite but the adult is free-living, is a common bridge between two worlds. Once a lineage adopts parasitism, it faces universal selective pressures that force unrelated organisms to evolve strikingly similar solutions. Whether a parasite is a single-celled protozoan or a multicellular worm, it must solve the same fundamental problems. It has to get from one host to another. It has to invade and survive inside the host, and it has to exploit host resources sustainably. Because there are only so many ways to do this successfully, almost all eukaryotic parasites have converged on one of 6 general strategies. The first of these strategies is that of the parasitoid. This is a strategy of maximum virulence, most famously employed by certain wasps and flies. A parasitoid typically lays its eggs in or on a single host individual. As the larvae grow, they consume the host from the inside out, eventually killing it. This is a relationship that sits on the border between parasitism and predation. Some parasitoids are idiobionts, meaning they paralyze their host immediately, essentially turning it into a fresh, living larder. Others are coenobionts, which allow the host to continue growing and moving, even regulating the host's hormones to ensure it doesn't pupate until the parasite is ready. The second strategy is that of the parasitic castrator. These organisms do not kill their host, as a dead host is a useless resource, but they completely destroy its ability to reproduce. By shutting down the host's reproductive system, the parasite diverts all the energy that would have gone into making offspring toward its own growth and the host's survival. A classic example is the barnacle genus Sacculina, which infects crabs. The barnacle grows a network of root-like structures throughout the crab's body, eventually taking over its hormonal control. An infected male crab will even develop female physical characteristics and begin to care for the barnacle's egg sac as if it were its own.
The third and fourth strategies involve how the parasite is transmitted. Directly transmitted parasites or trophically transmitted parasites. Directly transmitted parasites, like lice and mites, move from one host to another through physical contact. They typically spend their entire lives on a single host species and have evolved specialized structures to keep them anchored there. Trophically transmitted parasites, on the other hand, rely on being eaten. This strategy involves complex life cycles with 2 or more different host species. A fluke might start its life in a snail, then move to a fish, and only reach sexual maturity once that fish is eaten by a bird. The final 2 strategies are vector-transmitted parasitism and micropredation. Vector-transmitted parasites, like the agents of malaria, use a third party, usually a biting insect, to carry them from one vertebrate host to another. Micropredators, like mosquitoes, leeches, and ticks, are unique because they attack more than one host during their life. They are only in contact with any one host intermittently, taking a quick meal of blood and then moving on. This behavior makes them perfect vectors for smaller parasites, creating a nested hierarchy of exploitation.
The physical changes that accompany these strategies are often described as genomic or morphological regression, but that is a misleading term. While it is true that many parasites lose features they no longer need, such as eyes, wings, or complex digestive systems, they simultaneously evolve highly specialized innovations. For example, endoparasitic flatworms have undergone significant genomic reduction, shedding genes for basic metabolism because they can harvest those products from their host. However, they compensate with specialized tools. An endoparasite living in the constant, nutrient-rich environment of a host's gut doesn't need a complex brain, but it does need a rostellum with hooks or powerful suckers to prevent being flushed out. It needs a tough outer covering, or tegument, that is resistant to the host's digestive enzymes. Furthermore, many endoparasites have evolved the capacity for anaerobic respiration. Living in low-oxygen environments like the intestinal tract requires a specialized metabolism that can generate energy without a steady supply of oxygen. While they may lose traditional light-sensing organs, they evolve unique sensory innovations to navigate the host's body. Ticks, for instance, possess the Haller's organ, a complex sensory structure on their front legs that detects odors, heat, moisture, and vibrations, allowing them to pinpoint a passing host with incredible accuracy. The specificity of these adaptations is often tied to the host's own development, a phenomenon known as stadium specificity. Some parasites are only able to infect a host during a specific life stage. The tadpole pinworm, Girenicola batrachiensis, is a specialist of the amphibian gut. It thrives while the host is an herbivorous tadpole, but because its biology is so finely tuned to that specific environment, the parasite is lost the moment the host metamorphoses into a carnivorous frog. This specificity suggests that a host is not just a source of food, but a complex landscape of microenvironments. To survive in this landscape, parasites occupy distinct niches. Some are coelozoic, inhabiting the hollow spaces of organs like the gallbladder, the bladder, or the intestines. Others are histozoic, living deep within the host's tissues or even inside individual cells. These niches are so specific that closely related parasites can behave entirely differently depending on where they land. For example, the fluke Holopegus occidentalis shows remarkable niche preference. It may attach to the veins under a frog's tongue in one species, but reside only in the stomach of another. The sheer diversity of parasitic forms is a testament to the power of this strategy. Parasites are not just moochers in the sense of being lazy or simple. They are specialists that have honed their biology to exploit the most reliable resource on the planet, other living things. They have been humanity's companions throughout our entire evolutionary history. We carry heirloom parasites like head lice and certain pinworms, which have followed our ancestors since before we were even human, evolving alongside us as we migrated out of Africa. Others are souvenir parasites, which we picked up along the way as we moved into new environments and interacted with different animal species. These adaptations are not limited to the animal kingdom. Parasitism has also flourished among plants and fungi. About 1% of all flowering plants are parasitic. Some are hemiparasites, like mistletoe, which can still photosynthesize but steal water and minerals from their hosts. Others are holoparasites, like the dodder or the witchweed, which have lost their chlorophyll entirely. These plants have modified their roots into structures called haustoria, which penetrate the host's stem or roots to tap directly into its conductive system. Fungi have also mastered the parasitic lifestyle, often displaying a biotrophy-necrotrophy switch. Biotrophic fungi keep host cells alive for as long as possible to ensure a steady supply of nutrients, while necrotrophic fungi kill host cells and then feed on the remains. The belief that parasitism is an evolutionary dead end because it requires putting all your eggs in one basket is contradicted by the evidence of these ancient, thriving lineages. While a specialist parasite might face extinction if its host species disappears, the strategy itself is so robust that new parasitic lineages are constantly emerging to fill the void. Parasites act as regulators of ecosystems, controlling host populations and preventing any single species from dominating a habitat. They are a massive, though often invisible, component of food webs, sometimes making up more biomass than the top predators in an environment. By influencing host health and survival, parasites drive the evolution of their hosts in a constant, reciprocal dance. This dynamic relationship is the engine behind some of the most fundamental processes in biology, including the development of complex immune systems. As we look closer at this interaction, we find that the struggle to survive an infection has forced life to evolve one of its most complicated and costly traits, the ability to shuffle the genetic deck through sexual reproduction. In the natural world, survival is often compared to a race, but for a host and its parasite, it is a race where neither side ever truly reaches the finish line. This is the essence of the Red Queen hypothesis, a concept borrowed from the world of literature, where a character must run as fast as she can just to stay in the same place.
In biological terms, this means that a host must constantly evolve new defenses just to survive the latest onslaught of a parasite, while the parasite must simultaneously evolve new ways to bypass those defenses just to keep finding a meal. This eternal cycle of adaptation and counter-adaptation is known as a co-evolutionary arms race, and it is one of the most powerful engines of biological diversity on our planet. To understand how this dance begins, we have to look at the concept of negative frequency-dependent selection. Imagine a population of hosts with several different genetic profiles. A parasite will naturally find it easiest to infect the most common genetic profile because it provides the largest number of targets. As the parasite adapts to exploit this dominant group, the fitness of those common hosts plummets. Meanwhile, a rare genetic profile that the parasite hasn't learned to crack yet suddenly has a massive advantage. These rare individuals survive and reproduce more successfully, and over generations, their genetic profile becomes the new common one. The parasite is then forced to pivot, adapting to this new majority, which in turn favors a different rare group. This constant shifting prevents any single genetic type from dominating forever, maintaining a high level of genetic variation within the population. This pressure is so intense that many scientists believe it is the primary reason why sexual reproduction exists at all. Sex is a costly and complicated way to reproduce compared to simple cloning. You have to find a mate, risk being eaten or infected during the process, and you only pass on half of your genes. However, sex offers one incomparable advantage. It shuffles the genetic deck. Every offspring is a unique combination of genes, creating a population of moving targets that a parasite cannot easily pin down. A remarkable example of this can be found in the freshwater lakes of New Zealand, home to a small snail that exists in both sexual and asexual forms. In some populations, the snails are mostly clones, while in others they reproduce sexually. Researchers discovered a direct correlation between the prevalence of parasitic trematodes and the frequency of sex. In lakes where the water is thick with parasites that castrate the snails, the sexual snails thrive because their diverse offspring are more likely to have a combination of genes that resists infection. In areas with fewer parasites, the asexual clones, who don't have to waste energy finding mates, tend to take over. This shows that the threat of parasitism can actually force a species to maintain the complex machinery of sex.
The Red Queen's influence even extends to the social and mating behaviors of parasites themselves. Schistomas, the blood flukes responsible for schistosomiasis, provide a fascinating behavioral illustration of this hypothesis. While these parasites usually form permanent, monogamous bonds within the host's blood vessels, researchers have observed instances of schistome divorce. A female may leave her partner to mate with a more genetically distant male. This behavior is not random. By seeking out a partner with a different genetic background, the parasite decreases the risks of inbreeding and increases the genetic diversity of its offspring. This ensures that the next generation of flukes has a better chance of navigating the host's ever-evolving immune defenses. The specific battlegrounds where this genetic shuffling is most intense are well defined within the genomes of both hosts and parasites. In vertebrates, the most diverse regions of the genome are the major histocompatibility complex, MHC genes, which are responsible for recognizing foreign invaders. In plants, the equivalent battleground is found in resistance, R genes. Because of the balancing selection driven by parasites, these areas of the DNA are under constant pressure to maintain a vast array of variants. This diversity is the host's primary shield, ensuring that at least some individuals in a population will possess the molecular tools to recognize and fight off a new parasitic strain. We can actually see this arms race playing out over time by looking into the mud at the bottom of lakes. In a study of the water flea, Daphnia, and its bacterial parasites, researchers used the lake sediment as a biological time machine. Daphnia produce resting eggs that can stay dormant in the mud for decades, and their parasites produce similar resting stages. By taking cores of the sediment and reanimating both the hosts and the parasites from different layers, scientists could stage cross-time battles. They found that parasites were consistently most effective at infecting the hosts they lived with in their own time period. When exposed to hosts from the past or the future, the parasites were significantly less successful. This is definitive proof of the Red Queen in action. The parasites were constantly running to keep up with the contemporary defenses of their hosts, losing their edge against the older or newer versions. Sometimes the arms race produces what we call a heterozygote advantage, where carrying a bad gene is actually a shield against something worse. This is famously seen in the relationship between humans and malaria. The allele for sickle cell anemia causes a debilitating blood disorder in people who inherit 2 copies of it. However, those who carry just 1 copy of the gene, heterozygotes, possess red blood cells that are uniquely resistant to the malaria parasite, Plasmodium. In regions where malaria is a constant threat, this genetic variation is actively preserved by natural selection. The parasite exerts such a strong pressure that it keeps a potentially lethal human gene in circulation, because the protection it offers against the parasite outweighs the cost of the disease.
The molecular mechanisms behind these adaptations are incredibly sophisticated. In the Negev desert of Israel, researchers have tracked how Bartonella bacteria adapt to different species of wild rodents. They found that the bacteria use contingency loci, highly unstable parts of their genome consisting of simple sequence repeats. These segments act like rapid on-off switches for certain genes, such as those that produce adhesins, which the bacteria use to stick to host cells. By rapidly changing the length of these repeats, a population of bacteria can generate a massive variety of surface structures in a single generation. This allows them to stay one step ahead of the host's immune system, which is trying to recognize and attack those very structures. When the bacteria were moved between different rodent species, they saw these genetic switches flipping in real time, showing how quickly a parasite can retool itself for a new host. This genetic flexibility is not limited to bacteria. Even more complex parasites, like the single-celled Leishmania, use genetic exchange to fuel their evolution. For a long time, it was assumed that these parasites reproduced mostly by cloning themselves. However, recent genomic analysis has revealed that more than 70% of Leishmania isolates show evidence of genetic mixing and hybridization. By exchanging large chunks of genetic material through sexual reproduction, they can rapidly combine advantageous traits such as drug resistance or the ability to infect new types of insect vectors. The parasite's ability to adapt is often regulated by specific molecular triggers. In malaria parasites, the GDV1 gene has been identified as a master regulator of sexual conversion. This gene controls the rate at which the parasite switches from asexual replication within the blood to the production of sexual stages required for transmission to a mosquito. This is a specific molecular target of long-term adaptation. By fine-tuning the activity of this gene, the parasite can optimize its transmission strategy based on the host's immune status or the availability of vectors, ensuring it remains a viable competitor in the arms race. However, the benefits of sex and recombination are not always available to every parasite, and genetic architecture can impose significant constraints. In the case of the cuckoo finch, a brood parasite that lays its eggs in the nests of other birds, the evolution of egg mimicry is a double-edged sword. The genes for egg color and pattern are maternally inherited on the W chromosome. While this ensures that a daughter will inherit the same mimicry as her mother, the lack of recombination on that specific chromosome prevents the parasite from evolving new genetic combinations. This acts as a constraint, making it harder for the parasite to keep up with the host's own autosomal polymorphisms. The parasite is locked into a specific strategy, while the host has more freedom to shuffle its genetic deck, illustrating that the Red Queen's race is not always run on a level playing field. The broader distribution of biological diversity is often shaped by 2 different evolutionary paths: descent and colonization.
Descent, or co-speciation, occurs when a host and its parasite evolve together over eons, their lineages splitting in parallel like mirror images. This creates a highly specialized relationship where the parasite's history is inextricably linked to the host's. Colonization, on the other hand, involves host switching, where a parasite jumps to a new, often unrelated species. This distinction is fundamental to how the arms race drives diversity. While descent leads to deep specialized lineages, colonization allows parasites to exploit new niches, creating a messy and vibrant map of life that defies simple categorization. The arms race can also lead to a delicate balance between transmission and virulence. A parasite that kills its host too quickly is like a fire that burns its fuel too fast. It risks going out before it can spread to a new host. This often leads to the evolution of an optimal level of virulence, where the parasite's need to replicate is balanced against the host's need to survive long enough to facilitate transmission. This balance is often influenced by the environment, a concept known as the geographic mosaic theory of coevolution. Selection doesn't happen uniformly across a species range. Instead, you have hotspots where the interaction between host and parasite is intense and reciprocal, and cold spots where one side or the other has the upper hand, or where the interaction is missing entirely. In the islands of Finland, a species of perennial herb called Plantago lanceolata is frequently attacked by a powdery mildew fungus. Across thousands of small populations, the intensity of this battle varies wildly. In some areas, the plants have evolved high levels of resistance, while in others, the fungus has developed the tools to overcome those defenses. Gene flow between these populations, seeds blowing in the wind or fungal spores drifting across the landscape, constantly remixes these traits, ensuring that neither the plant nor the fungus ever wins the war on a regional scale. The presence of other parasites can also change the trajectory of the arms race. We often think of parasites competing to see who can be the most specialized, but competition can actually favor the evolution of generalists. In the study of feather lice on doves, researchers found that when multiple species of lice are competing for the same space and resources on a single bird, they are more likely to evolve the ability to switch to new host species. Specialization is often seen as an evolutionary dead end because a specialist is doomed if its host goes extinct. However, the presence of competitors creates a pressure to find empty hosts, driving the parasites to become generalists who can survive on a variety of different birds. This challenges the traditional view that evolution always moves toward greater and greater specialization. The arms race can even involve physical structures that seem almost too strange to be real. Consider the rough-skinned newt and the common garter snake. The newt produces tetrodotoxin, a neurotoxin so potent that a tiny amount can paralyze and kill a large predator. But the garter snake has evolved a countermeasure, a specific mutation in the sodium channels of its nerve cells that prevents the toxin from binding. This allows the snake to eat the newt and survive. In response, the newts have evolved to produce even higher levels of toxin, leading to a situation where some newts carry enough poison to kill dozens of humans. This level of toxicity is far beyond what is needed to deter any other predator. It exists solely because of the snake's resistance. In areas where the snakes are not present, the newts produce much lower levels of toxin, proving that the cost of producing such a weapon is only worth it when you are locked in a life-or-death struggle with a resistant foe. These interactions show that parasites are not just passive passengers. They are active participants in the shaping of life's history. They drive the creation of new genes, the maintenance of sexual reproduction, and the distribution of species across the globe. The Red Queen's dance ensures that the biological world remains in a state of constant flux, with every new defense triggering a new offense in an endless cycle of innovation. This process of reciprocal change is what allows life to remain resilient in the face of a changing world, as both host and parasite are forced to keep running just to stay in the game. The complexity of these relationships is perhaps most evident when the parasite stops being a simple predator and starts acting as a puppeteer, taking control of the host's very actions to ensure its own survival.
Chapter 3. The Extended Phenotype. How Parasites Pull the Strings.
For more than a century, naturalists have been fascinated and often unsettled by behaviors in the animal kingdom that seem to defy the logic of self-preservation. We see ants that climb to the highest tips of grass blades and clamp their jaws in a permanent death grip, crickets that hurl themselves into water to drown, and snails with eye stalks that pulse with vivid flashing colors like miniature neon signs. These are not biological accidents or signs of madness in the host. Instead, they are the visible expressions of the adaptive host manipulation hypothesis. This concept suggests that a parasite can evolve to seize control of its host's nervous system or physiology, turning the host into a vehicle designed to maximize the parasite's own transmission. In these scenarios, the host's body becomes what scientists call an extended phenotype of the parasite. The genes of the parasite are essentially pulling the strings of the host's behavior from the inside. As established, the single-celled protozoan Toxoplasma gondii must return to a feline host to reproduce, a requirement that has driven the evolution of a phenomenon known as fatal attraction. In a healthy state, a mouse has an innate, life-saving fear of the smell of cat urine. However, once infected with toxoplasma, that fear is not just dampened, it is replaced by a strange attraction. The infected mouse will actively seek out areas where cats have marked their territory. By making the mouse less cautious and more likely to be eaten by a feline predator, the parasite ensures it reaches its final destination.
This manipulation is incredibly precise, targeting specific neural pathways associated with fear and arousal. Interestingly, this effect isn't limited to rodents. In studies of larger mammals, such as chimpanzees, infected individuals show a similar loss of avoidance behavior toward the urine of their natural predators, like leopards. Even in humans, where the parasite is often considered to be in a latent or dormant state, researchers have observed subtle shifts in personality and behavior. Infected men have been found to be more risk-taking and aggressive. Perhaps most grisly is the discovery that infected humans find the scent of cat urine significantly less unpleasant than uninfected individuals do, with some even finding the odor attractive. These changes suggest that the mechanisms Toxoplasma uses to manipulate its hosts are ancient and deeply embedded in the shared biology of mammals. The manipulation of behavior is equally sophisticated in the world of vector-borne diseases. Malaria, caused by the parasite Plasmodium falciparum, employs a dual strategy of manipulation to ensure its spread. First, the parasite alters the body odor of infected humans, making them more attractive to foraging mosquitoes. This ensures the parasite is picked up by its next vector. Once inside the mosquito, the parasite changes the insect's behavior as well. Infected mosquitoes become more persistent and bite more people per night than their uninfected counterparts. By increasing the frequency of contact between the vector and new potential hosts, the parasite maximizes its chances of transmission.
While some parasites use chemical signals to lure their hosts toward danger, others use elaborate physical deception. The freshwater mussel Lampsilus ventricosa has evolved a remarkable behavioral lure to ensure its larvae reach their fish hosts. The mussel possesses a mantle flap that it undulates in the water to perfectly mimic the appearance and movement of a small minnow. When a predatory fish, such as a bass, strikes at the minnow, the mussel releases a cloud of larvae that attach to the fish's gills. An even more aggressive lure is seen in the mite Histiostoma laboratorium. This parasite lives on the surface of mushrooms and waits for a fruit fly to land. When a fly approaches, the mite launches itself like a surface-to-air missile, latching onto the fly to be transported to a new environment. The evolution of such complex control usually occurs in parasites with a trophic mode of transmission, where the parasite must move from a prey animal to a predator. Consider the hairworm Spinochordotus tellini, which infects crickets. The adult hairworm lives in water, but its juvenile stage develops inside the body of a terrestrial cricket. To get back to the water, the worm must force the cricket to leave its dry habitat.
As the worm reaches maturity, it manipulates the cricket's brain, causing the insect to seek out a body of water and jump in. As the cricket drowns, the adult worm, which can be several times longer than its host, erupts from the cricket's abdomen to begin its aquatic life. This is a high-stakes gamble. If the cricket doesn't find water, the worm cannot reproduce. Consequently, the selection pressure to be an effective puppeteer is immense.
Another striking example is the trematode Neoleucochloridium, often called the lighthouse snail parasite. This fluke infects the digestive systems of birds, which release the parasite's eggs in their droppings. Snails then consume these eggs. Once inside the snail, the parasite migrates to the eye stalks and forms large pulsating sacs filled with larvae. These sacs are brightly colored and move in a way that mimics the appearance of a wriggling caterpillar. But the manipulation goes deeper than just a physical disguise. The parasite also alters the snail's light-sensing behavior. Snails normally prefer dark, damp places to avoid predators, but an infected snail will crawl out into the open, brightly lit parts of a plant. There, its pulsing eye stalks act as a beacon, attracting the attention of birds. The bird eats the eye stalk, and the parasite successfully transitions to its next host. While these examples in animals are dramatic, host manipulation is just as prevalent in the world of plants. Plant-infecting viruses and bacteria have evolved sophisticated ways to control both their host plants and the insects that act as their vectors. For example, the tomato spotted wilt virus changes the behavior of the thrips that carry it. Infected thrips become more aggressive feeders, biting more plants than their uninfected counterparts, which directly increases the spread of the virus. In many cases, plant pathogens create a fatal attraction of their own. Viruses like the barley yellow dwarf virus or the potato leaf roll virus can change the chemical signals emitted by an infected plant. These plants begin to produce volatile organic compounds that are irresistible to aphids. To an aphid, an infected plant might smell or look like a higher quality food source than a healthy one. Once the aphid lands and begins to feed, it picks up the virus. In a clever secondary twist, some viruses then change the aphid's preference. Once infected, the insect is more likely to be attracted to the scent of a healthy plant, ensuring the virus is delivered to a new host. Some bacteria, like Canditatus Liberibacter asiaticus, which causes citrus greening disease, use a similar strategy with psyllids, creating a cycle of attraction and dispersal that can devastate entire orchards. This level of control requires the parasite to interfere with the most fundamental processes of the host. One fascinating mechanism involves the barber's pole worm, a blood-feeding nematode that infects livestock. This parasite has evolved a way to hijack the host's own hormones. Specifically, it uses a receptor to exploit host insulin, using the host's metabolic signals to drive its own infection process and reproduction. By tapping into the host's insulin signaling, the parasite can synchronize its development with the nutritional state of the host. Most remarkably, the parasite uses these signals to shift the sex ratio of its offspring toward females, ensuring a higher reproductive output when resources are plentiful. The ability of a parasite to manipulate its host often raises a difficult question. How can we tell the difference between a parasite's manipulation and a host's own adaptive response to being sick? Consider the sneeze. When we sneeze, are we trying to expel a pathogen as part of our immune defense, or is the germ making us sneeze to help itself travel to a new person? This conflict is central to understanding sickness behavior. Mammals respond to infection with fatigue, fever, and a loss of appetite. If a sneeze is a host defense, it is an attempt to clear the airways. However, if the sneeze is a parasite's tool, it is a high-velocity delivery system for transmission. A clever parasite might evolve to exploit these very responses. If a parasite needs its host to stay still and be eaten, it might amplify the host's natural fatigue. If it needs the host to be active and social to spread, it might suppress the sickness behavior entirely, ensuring the host continues to sneeze in public spaces rather than resting in isolation.
This conflict is why host manipulation is rarely perfect. It is an ongoing evolutionary struggle. The host is under constant pressure to evolve resistance to being manipulated, while the parasite must find new ways to bypass those defenses. This might explain why we see a surprising lack of reported host manipulation in many human sexually transmitted diseases. Because STDs are already guaranteed a high probability transmission event, the act of mating, they may not need to evolve the elaborate behavioral controls seen in parasites that rely on chance encounters. However, there are notable exceptions in the animal kingdom. The iridovirus, IIV6/ARVA, in crickets provides a perfect counterexample. It makes infected males significantly more sexually attractive to healthy females, facilitating the transmission of the virus through the very act of mating. The genetic toolkit for this puppetry is becoming clearer through the study of parasite genomes. Researchers have identified specific gene families that are expanded in manipulating parasites. In single-celled parasites like Trypanosoma, these include quirky molecular biology traits such as genes for extensive RNA editing. These traits allow the parasites to bypass standard eukaryotic textbook models of gene expression, giving them the flexibility to survive in diverse host environments and interfere with host cell signaling. The broader genetic toolkit of such complex worms allows them to secrete a variety of proteins that can cross the blood-brain barrier or mimic host signaling molecules. The evolution of manipulation also shows a remarkable degree of convergence. Unrelated parasites have hit upon the same behavioral buttons to push in their hosts. Multiple different species of fungi, viruses, and even parasitic insects have evolved ways to make their hosts seek out high ground before they die. This summit disease phenomenon represents another of the adaptive peaks discussed in earlier chapters. By forcing the host to climb, the parasite ensures that its spores or offspring will be dispersed over a wider area by the wind or by falling onto other hosts below. Whether it is a zombie ant or a zombie caterpillar, the end result is the same, achieved through different molecular pathways but driven by the same ecological necessity. We are also beginning to see how manipulation can be influenced by the presence of multiple parasites within a single host. In some cases, different parasites might have conflicting interests. One might need the host to be active, while another needs it to be dormant. This can lead to a tug-of-war over the host's behavior. In other cases, parasites might work together. A primary parasite might suppress the host's immune system, making it easier for a secondary, manipulating parasite to take hold. This complex web of interactions suggests that the puppeteer is often part of a much larger ensemble cast. Understanding how parasites manipulate host physiology and behavior has profound implications for medicine and agriculture. If we can identify the specific receptors or signaling molecules a parasite uses to hijack a host, we can develop targeted treatments to break that control. For example, by blocking the receptor that the barber's poleworm uses to sense host insulin, we could potentially stop the infection without needing to use toxic chemicals. In the case of vector-borne diseases like malaria, understanding how the parasite makes humans more attractive to mosquitoes could lead to new ways of masking those signals and reducing transmission rates. These puppeteers remind us that the boundary between two organisms is often more porous than it appears. A host is not just a home for a parasite. It is a resource to be managed, a vehicle to be steered, and a tool to be used. The evolution of host manipulation represents one of the most sophisticated levels of biological interaction, where the survival of one species depends on its ability to rewrite the very nature of another. This genetic control of behavior is a testament to the power of natural selection to find and exploit any possible advantage, no matter how strange or grisly it may seem to us. The same genetic mechanisms that allow a parasite to control a host's actions can also be repurposed over millions of years to build entirely new biological structures, a process that has left its mark deep within the mammalian genome. Chapter 4: Architects of the Womb: The Placenta's Parasitic Origins.
When we think of evolution, we often imagine a slow, steady accumulation of small changes within a single species. But some of the most profound leaps in the history of life have been driven by a much more radical process, the integration of one organism into another. We have already seen how parasites can act as puppeteers, controlling the behavior of their hosts from the outside or the inside. But there is an even deeper level of interaction. Over millions of years, some parasites have moved so far into the interior of their hosts that they have become part of the host's own genetic and cellular machinery. These hidden architects, ranging from mobile genetic elements within our DNA to bacterial endosymbionts living inside animal cells, have played a decisive role in building the complexity we see in the natural world today. Perhaps the most stunning example of this is the evolution of the mammalian placenta, a structure that owes its existence to what are essentially genomic parasites. To understand this, we have to look at transposable elements, often called jumping genes. These are stretches of non-protein coding DNA that have the unique ability to change their position within a genome. For a long time, these were dismissed as junk DNA, or genomic parasites, because they seemed to serve no purpose other than to replicate themselves, sometimes at the expense of the host organism. However, we now know that these elements are powerful drivers of evolutionary innovation. In the early history of mammals, an invasion of these genomic parasites coordinated a massive genome-wide reorganization that allowed for the development of the uterus and the placenta. Before this transition, the ancestors of mammals laid eggs, relying on a yolk to provide all the nutrients a developing embryo needed. As mammals evolved toward live birth, they had to solve a major biological problem: how to maintain a prolonged pregnancy where the fetus receives nutrients directly from the mother. This required a new way for the mother and fetus to communicate, and, crucially, a way to prevent the mother's immune system from attacking the fetus. From the perspective of an immune system, a fetus is a foreign object, carrying half of its genes from the father. Without intervention, the mother's body would recognize the fetus as an invader and attack it exactly like a parasite. Researchers have found that thousands of genes evolved to be expressed in the uterus of early mammals to facilitate this communication and immune suppression. Remarkably, these genes were not created from scratch. Instead, they were existing genes that previously functioned in other parts of the body, such as the brain, the digestive system, or the circulatory system. The force that recruited these genes and moved them into the service of the uterus was the jumping gene. These transposable elements carried with them regulatory sequences, specifically binding sites for the hormone progesterone. By randomly inserting themselves near existing genes, these genomic parasites essentially brought the instructions for when and where those genes should be turned on. This allowed a single signal, the release of progesterone, to activate a massive network of genes across the entire genome, creating the complex environment necessary for a placenta to function. One of the most critical sets of genes repurposed in this way were those responsible for suppressing the local immune response in the uterus, effectively taming the mother's defenses so the developing life could survive. This theme of a parasite becoming an essential architect of its host's life is repeated at the cellular level with endosymbionts. These are bacteria that live inside the cells of another organism, and their relationships range from purely parasitic to obligately mutualistic. The genus Wolbachia is perhaps the most widespread and successful of these internal residents. Found in up to 70% of all insect species, as well as in many spiders, mites, and parasitic worms, Wolbachia is a master of reproductive manipulation. It ensures its own survival by favoring the production of infected females, the only sex that can pass the bacteria on to the next generation. Wolbachia employs several grisly strategies to achieve this. It can cause male killing, where infected males die during their larval development, leaving more resources for their sisters. It can induce feminization, turning genetic males into functional females. In some species, it triggers parthenogenesis, allowing females to reproduce without ever needing a male.
Perhaps most common is cytoplasmic incompatibility, where infected males cannot successfully reproduce with uninfected females. This creates a powerful selective sweep. Because infected females can mate with any male, they quickly outcompete uninfected females until the entire population carries the bacteria.
While these behaviors are clearly parasitic, in other lineages, the relationship has shifted into one of total dependence. In the filarial nematodes that cause diseases like elephantiasis and river blindness, Wolbachia has become an obligate endosymbiont. The worms have lost the ability to produce certain chemicals necessary for their own reproduction and survival, and they now rely entirely on the bacteria to provide them. This dependency is so absolute that the most effective way to treat these worm infections in humans is not to target the worm itself with toxic drugs, but to give the patient a simple course of antibiotics. When the Wolbachia dies, the worm becomes sterile and eventually dies as well. The Parasites parasite has become the host's life support system. A similar nested dependency is found in ticks. These blood-sucking arachnids have a diet that is extremely high in protein and iron, but dangerously low in essential vitamins, particularly B vitamins. To solve this nutritional challenge, ticks have evolved obligate relationships with bacterial endosymbionts from the genera Coxella and Francisella. These bacteria are passed from a mother tick to her offspring through her eggs. They live inside specialized cells and act as miniature vitamin factories, synthesizing biotin, riboflavin, and folate that the tick cannot get from blood alone. Genomic analysis of these endosymbionts reveals a classic hallmark of this lifestyle: massive genome reduction. Because the bacteria live in the stable, nutrient-rich environment of the tick's body, they have shed most of the genes they would need to survive in the outside world, effectively outsourcing their own basic metabolism to their host.
This trend reflects the universal tendency for organisms in stable, nutrient-rich host environments to shed redundant genetic systems and outsource metabolic functions, a pattern of genomic reduction and specialization also seen in endoparasitic flatworms. Even the most fundamental structures of the cell have been shaped by these ancient interactions. The development of the nucleus itself, the defining feature of all complex life, may have roots in this kind of coevolutionary molding. By studying the molecular biology of trypanosomes, parasites responsible for sleeping sickness, researchers have uncovered a unique architecture in their nuclear pore complexes. These are the channels that control the flow of information into and out of the nucleus. In most animals, plants, and fungi, these pores are asymmetric, with different proteins on the inside and outside faces to help establish the direction of transport. In trypanosomes, however, the pore is almost completely symmetric. This discovery supports the protocodomer hypothesis, which suggests that the nuclear envelope and the nuclear pore share a common ancestor with the simple protein coats that form on other membrane-bound structures within a cell. The trypanosome's simple, symmetric pore may represent a primitive version of this structure that existed before the last common ancestor of all eukaryotes. These parasites, because they diverged from our own lineage more than 1 billion years ago, act as living fossils that reveal how the very first complex cells were put together. Their molecular biology, including specialized mechanisms like RNA editing, highlights the diverse solutions forged in parasitic interactions. The integration of these hidden architects into our biology shows that the line between self and other is often a historical illusion. We are composite creatures, our genomes littered with the remnants of ancient viruses and jumping genes, our cells sometimes dependent on the products of internal bacteria. This deep history of colonization and cooperation has allowed life to bypass the slow process of incremental change, instead jumping to new levels of complexity by absorbing the innovations of others. This process has not stopped. It continues today in every ecosystem on Earth, as parasites and hosts continue their intimate dance of adaptation. But as the world enters a new era of rapid, human-driven change, these ancient relationships are being tested in ways they have never been before. Chapter 5.
The Anthropocene, Rapid Evolution and the Warning. The human footprint is now visible in every corner of the Earth, defining a new geological epoch known as the Anthropocene. In this era, the biotic and abiotic conditions of our planet are shifting at a pace that far outstrips the natural background rate of change. While we often focus on the plight of charismatic megafauna or the bleaching of coral reefs, an equally profound and perhaps more consequential transformation is occurring within the world of parasites. Building on their inherent capacity for rapid evolution, parasites are not merely passive victims of environmental change. They are dynamic participants, responding to human-induced pressures with new adaptive directions that could reshape the future of global health, food security, and ecosystem stability.
The raw material for this rapid evolution is already present in the form of substantial standing genetic variation. Parasite populations often harbor a vast reservoir of genetic diversity that allows them to pivot almost instantly when a new pressure is applied. We see this most clearly in the intensive environments of livestock production and aquaculture, where humans have inadvertently created massive evolutionary laboratories. The widespread use of anthelmintics has exerted a selective pressure so intense that drug resistance is now a global crisis. By employing whole genome resequencing of archive samples and conducting all-against-all cross-infections in aquaculture facilities, scientists are beginning to map the precise genetic changes that allow pathogens to thrive under intensive management. When a farmer treats a herd or a tank with a specific drug, the few individual parasites that happen to possess a resistant mutation survive and multiply, rendering once effective treatments useless in just a few seasons. This human-induced evolution isn't limited to drug resistance. Our attempts to control diseases through vaccination can also alter the evolutionary trajectory of parasites. In some systems, vaccines that reduce the symptoms of a disease but do not stop its transmission can create an environment where more virulent strains of the parasite are favored. If a parasite is no longer held back by the risk of killing its host too quickly, because the host is partially protected by a vaccine, the parasite can evolve to replicate more aggressively. This leakiness in control measures forces us to recognize that the parasites themselves are changing in response to our interventions, often becoming more formidable in the process. The marine environment offers some of the most striking evidence of how human activities and environmental shifts are altering parasite dynamics over decades. Researchers recently turned to an unconventional source to track these changes: old cans of salmon. By opening nearly 200 cans of fillets collected over a 42-year span from the Gulf of Alaska and Bristol Bay, scientists were able to conduct a retrospective autopsy of the ocean's health. They focused on tiny parasitic worms known as anisakids, or sushi parasites. These are the same organisms that cause anisakiasis in humans if consumed alive in raw fish, though the worms in the cans were long dead. Because these parasites have complex life cycles that require multiple hosts, moving from krill to small fish, then to larger fish like salmon, and finally to marine mammals like seals or orcas, their presence serves as a biological barometer for the entire food web. The study revealed a surprising and nuanced trend. Levels of these parasitic worms increased significantly in chum and pink salmon between 1979 and 2021, yet remained steady in coho and sockeye. This species-specific divergence highlights the complexity of the Anthropocene. Different anisocket species rely on different host combinations, meaning a recovery for one part of the food web does not guarantee a universal shift for all parasites. To an ecologist, the rise in chum and pink salmon is a sign of a recovering ecosystem, likely reflecting the success of the Marine Mammal Protection Act of 1972. More marine mammals mean more opportunities for the parasites to complete their life cycles. This underscores a fundamental truth. Human policy and conservation efforts can have cascading effects that reach all the way down to the microscopic level of a parasite's reproductive success.
However, the news is not all positive. Climate change is introducing a level of environmental unpredictability that many parasites may struggle to survive. Rising water temperatures impose a dual penalty on many host parasite systems. In aquatic environments, thermal stress can confer a critical advantage to parasites with direct life cycles by accelerating their development and increasing their infective capacity. At the same time, this stress can severely compromise the immune systems of the fish they infect. This toxic synergy has been linked to massive fish mortality events in both wild populations and global aquaculture.
The specificity of these thermal responses is captured in thermal performance curves. In a warming world, we are seeing increasing instances of thermal mismatch, where a host and its parasite have different optimal thresholds. For example, in the relationship between the blue crab and its dinoflagellate parasite, the parasite's optimal growth occurs at temperatures that simultaneously cause the crab's innate immune response to decline. When the environment shifts just a few degrees, the balance of power tips decisively in favor of the pathogen. While we often expect species to move poleward in response to warming, some organisms demonstrate more complex, nonlinear range shifts. A long-term study on mountain white pine blister rust found that warming moved the disease's optimal conditions upslope. This actually reduced the prevalence of the rust in dry lowlands while simultaneously increasing its impact in cooler, high-elevation zones. This proves that the poleward march is not the only path for a changing climate. Parasites will track their optimal niches wherever they move, often with devastating consequences for high-altitude refugia. In the Arctic, where climate change is occurring more rapidly than anywhere else on Earth, the stakes are particularly high. Parasitic nematodes in these regions have evolved sophisticated adaptations to survive extreme cold. But as the Arctic thaws, the windows for transmission are changing. Warmer, longer summers are accelerating the development of parasites like the reindeer brainworm within its snail intermediate hosts. This leads to earlier and more severe outbreaks that can devastate both wild caribou herds and the semi-domesticated reindeer populations that are essential to pastoral communities.
While some parasites are thriving and expanding their ranges, many others are facing an extinction crisis. Conservative models suggest that between 5 and 10% of all parasite species may be committed to extinction by the year 2070 due to climate-driven habitat loss alone. When we account for co-extinction, the process where a parasite disappears because its host species has gone extinct, that number could rise to as high as 30% for groups like parasitic worms. To track these silent disappearances, researchers have developed the Parasite Extinction Assessment and Red List, or PARL. This online atlas provides a concrete framework for monitoring the health of parasite populations, moving beyond generic observations to provide a data-driven map of which species are most at risk. The loss of parasite biodiversity is not just a matter of losing moochers. Parasites play a vital role in regulating ecosystems and maintaining the health of host populations. They act as connectors in food webs and can even serve as a buffer against the emergence of more virulent, invasive pathogens. When we alter the environment through pollution, deforestation, or urbanization, we disrupt these ancient relationships. In Cameroon, for example, the deforestation of tropical rainforests allowed a species of human-infecting fluke to move into areas previously occupied by a different, less invasive species. The two species hybridized, leading to a new, more competitive strain that altered the local dynamics of disease.
This pattern of hybridization and range shifting is a recurring theme. As we move species across the globe, we are facilitating novel host parasite assemblages. Invasive species often succeed because they have escaped their native parasites, but they can also bring hitchhiker parasites with them. At the invasion front of cane toads in Australia, researchers have documented the emergence of superinfective lungworms. These parasites have evolved at a breakneck pace to exploit the rapidly expanding toad population, demonstrating how biological invasions can trigger intense evolutionary arms races in real time. Even our technological innovations are being integrated into these evolutionary battles. In the world of aquaculture, the rise of bacterial diseases has led to the exploration of phage therapy, the use of viruses that eat bacteria. However, the bacteria are not defenseless. They possess CRISPR-Cas systems, an adaptive immune system that allows them to remember and cut the DNA of invading phages. Long-term studies in fish farming facilities have shown these two microscopic forces locked in a constant arms race. As the bacteria evolve new spacers in their CRISPR arrays to recognize the phages respond by expanding their genome size or mutating their proteins to evade detection.
The reach of the parasitic lifestyle has even extended into our digital lives, providing a modern lens through which to view human evolution. Some researchers have argued that the relationship between humans and smartphones has transitioned into something resembling parasitism. These devices parasitize our time, attention, and personal information to serve the interests of technology companies and advertisers. The algorithms in our pockets are designed to nudge our behavior, to keep us scrolling and clicking, mirroring the way a biological parasite nudges its host's behavior to favor its own transmission. We have become so dependent on these digital extensions of our minds that we suffer from mood disorders, sleep loss, and weakened offline relationships, the tangible costs of this modern interaction. This modern analogy highlights a key feature of all parasitic relationships, the difficulty of detection and response. In the natural world, a host must be able to detect exploitation before it can evolve a defense. In the Anthropocene, the complexity of our environmental and technological impacts makes it difficult to see the evolutionary shifts until they are well underway. We are seeing a redistribution of parasite biodiversity that is both unpredictable and unprecedented. In some places, parasite richness may increase by an order of magnitude as species track shifting climates. In others, we may see a collapse of complex transmission chains.
In 2026, a group of leading parasitologists issued a formal scientist's warning on the rapid evolution of parasites in the Anthropocene. This document, mirroring the grave warnings issued for climate change and general biodiversity loss, serves as a call to action. It acknowledges that we are no longer just observers of parasite evolution, we are its primary drivers. As we continue to reshape the Earth's chemistry, climate, and connectivity, we are effectively rewriting the rules of the game for every living thing. The parasites, with their ancient history of colonization and their mastery of the mooching strategy, are proving to be some of the most resilient and responsive authors of this new chapter. As we move forward, we must ask, are we prepared for the new generation of specialists we are inadvertently designing? And what role will these rapidly evolving architects play in the ecosystems of tomorrow?
The evolution of parasites reveals that life is not a solo journey, but an interconnected dance of exploitation and innovation. From the ancient origins of the placenta to the zombie puppeteers of the insect world, parasites are masterful architects of biological diversity. They have driven the evolution of sexual reproduction, shaped our immune systems, and are now rapidly adapting to the human-altered landscapes of the Anthropocene. We must recognize that the line between host and parasite is often a historical illusion. We are composite creatures shaped by those who eat at our table. As parasites continue to regulate ecosystems and respond to our changing world, they remind us that no species exists in isolation. As we reshape the planet, we must wonder, are we truly the masters of our environment, or simply the latest hosts in an eternal, evolving race? Thank you for listening.